This interactive video is a quick review of the reactions involved in the utilization of glucose to produce energy in the form of ATP. It is meant to solidify your knowledge of these key reactions. You will be prompted to answer questions throughout the presentation.
Glycolysis is my favorite reaction. It is performed in the cytoplasm of almost every living organism on the planet to make ATP. I have to say almost because some variance on this set of reactions was reported in 2012. Glycolysis, as the name implies, is the splitting of glucose. The priming reactions begin with the addition of two molecules of ATP, one at a time, which produces a 6-carbon sugar with 2 phosphates attached to it, fructose 1,6-bisphosphate. The cleavage step splits the 6-carbon molecule into 2 3-carbon molecules, each with 1 phosphate. Now we need to charge up these molecules with more phosphates, but if we keep using ATP, we'll never end up producing it, which is the whole point of glycolysis. So now an enzyme will attach inorganic phosphate to the 3-carbon molecule. This enzyme requires the reduction of NAD+. Now we can pluck off the phosphates and stick them onto ADP to form ATP. We do this twice. And we are left with pyruvate, a 3-carbon molecule with no phosphates. So we began by using 2 ATPs, but we ended up producing 4 for a net gain of 2 ATPs, and voila, life goes on. We're left with one problem though. We can eat more sugar, but we've used up our coenzyme, NAD+. How are we going to resolve that? Stay tuned. Now pause this video and practice drawing glycolysis. If you can, teach the process to someone willing to listen. The simplest way to recycle the coenzymes so that we can keep doing glycolysis is fermentation. During lactic acid fermentation, such as that which occurs in bacteria or in muscle cells, the pyruvate that resulted from glycolysis is hydrogenated into lactate by dehydrogenating NADH. Now we have our NAD+ back. Notice no extra ATP is produced. This reaction happens once for each pyruvate generated by glycolysis. During alcohol fermentation, which occurs in yeast cells, the pyruvate that resulted from glycolysis is converted into carbon dioxide and ethanol, a 2-carbon molecule with the hydrogen taken from the NADH. And now, once again, we have our NAD+ back. This reaction happens once for each pyruvate generated by glycolysis. Now stop the video and practice drawing both fermentation reactions. Glycolysis and fermentation only produce 2 molecules of ATP out of 1 molecule of glucose. This is because there is still plenty of energy remaining in the bonds of lactate or ethanol, as evidenced by the fact that the latter can burn. In order to release all of the energy in glucose, it must be combusted. It then releases 686 kilocalories per mole. Of course, living organisms can't live if they burn up, but they can accomplish the complete oxidation of glucose in the small controlled steps of aerobic respiration. Aerobic respiration, the complete oxidation of glucose, takes place in several steps. These include glycolysis, the conversion of glucose into a pyruvate that results in the production of ATP and NADH, pyruvate oxidation, which forms acetyl-CoA along with carbon dioxide and NADH, the Krebs cycle, which results in the formation of carbon dioxide, ATP, NADH and FADH2, the electron transport chain, which removes hydrogen and electrons from NADH and FADH2 and creates a proton concentration gradient across the inner membrane of the mitochondrion and passes the electrons along a series of protein complexes until they ultimately combine with oxygen and hydrogen ions to form water. And chemiosmosis, which results in the activation of ATP synthase and the production of a whole lot of ATP. Glycolysis occurs outside of the mitochondria in the cytoplasm. Pyruvate oxidation occurs in the intermembrane space of the mitochondria. The Krebs cycle occurs in the mitochondrial matrix, and the electron transport chain occurs on the inner mitochondrial membrane. Now let's take a closer look. Glycolysis in aerobic respiration is the same for all organisms, so let's proceed to the oxidation of pyruvate. Pyruvate oxidation occurs in the intermembrane space of the mitochondria. Pyruvate is converted into acetyl coenzyme A. This requires the investment of NAD+ and coenzyme A. This reaction forms NADH and carbon dioxide. The Krebs cycle occurs within the matrix of the mitochondria. Watch this video to see what becomes of the pyruvate after it has been oxidized to acetyl coenzyme A. By the way, most people abbreviate it as acetyl-CoA, not acetyl-CoA, as the narrator pronounces it.
The 2-carbon acetyl portion of the acetyl-CoA is transferred to a 4-carbon molecule, producing a 6-carbon compound. The CoA carrier molecule is released. Carbon dioxide is then released from the 6-carbon molecule, forming a 5-carbon compound. In this step, hydrogen is removed and transferred to NAD+ to form NADH. Next, a second oxidation and decarboxylation occurs. Again, NADH and carbon dioxide are produced. In addition, a molecule of ATP is produced. As a result of these reactions, a 4-carbon molecule is formed in the Krebs cycle. Finally, the 4-carbon molecule is further oxidized, and the hydrogens that are removed are used to form NADH and FADH2. These reactions regenerate the 4-carbon molecule that initially reacts with acetyl-CoA. Each glucose molecule is broken down into 2 pyruvate molecules during glycolysis. Then each pyruvate is converted to acetyl-CoA, which enters the Krebs cycle. Thus, for each glucose molecule, the Krebs cycle must complete 2 circuits to completely break down the 2 pyruvate molecules.
Now stop the video and practice drawing the Krebs cycle. The electron transport chain is a series of electron carrier proteins embedded in the inner mitochondrial membrane. Here, the electrons from NADH and FADH2 that were formed during glycolysis, the oxidation of pyruvate in the Krebs cycle, are removed and passed along a chain of electron carriers. When some of these proteins receive the electrons, they pump protons from across the inner mitochondrial membrane from the matrix to the intermembrane space. The electrons keep moving from one protein complex to another until they reach the final electron acceptor, which is oxygen. The proton gradient created by the electron transport chain is not rectified by diffusion because the inner mitochondrial membrane is not very permeable to hydrogen ions. Most of the protons pumped into the intermembrane space can only diffuse out through a specialized channel associated with an ATP synthase. When this enzyme is activated, it phosphorylates ADP to make ATP. Let's take a closer look at the electron transport chain and chemiosmosis. Remember, these processes occur on the inner mitochondrial membrane. When NADH enters the electron transport chain, electrons and hydrogens are released from NADH, and we finally get our NAD+ back. When the electrons get to the first proton pump, it shoots a hydrogen ion into the intermembrane space. The electrons keep going through complex Q and onto the second proton pump, which shoots a second hydrogen ion into the intermembrane space. The electrons keep going through complex C and onto the third proton pump, which shoots a third hydrogen ion into the intermembrane space. The final electron acceptor is oxygen, which then picks up two hydrogen ions and becomes water. The electrons transferred from protein to protein drive an increase in the hydrogen concentration within the intermembrane space. These hydrogens diffuse back through the ATP synthase protein, resulting in the formation of ATP, the process known as chemiosmosis. So, for every NADH entering the electron transport chain, 3 ATPs are made. When FADH2 enters the electron transport chain, it releases 2 hydrogens and 2 electrons, and we get our FAD back. The electrons skip the first proton pump and are donated to complex Q. The electrons move to the second proton pump, which shoots a hydrogen into the intermembrane space. The electrons continue on to complex C and then to the third proton pump, and a second electron is pumped into the intermembrane space. So, for every FADH2 that enters the electron transport chain, 2 ATPs are made. Now stop the video and practice drawing the electron transport chain and chemiosmosis. If you were wondering how ATP synthase works, here's how. ATP synthesis is carried out by a tiny rotary motor driven by the hydrogen ion gradient. It consists of a transmembrane domain and a stalk and knob complex that has enzymatic activity. Protons travel through a channel in the transmembrane domain, which causes the stalk and knob to rotate. Mechanical energy changes the conformation of the catalytic domain, and the enzyme ATP synthase enables a phosphate group to be dehydrated onto ADP to yield ATP. Aerobic respiration consists of glycolysis, which is the conversion of glucose into 2-pyruvates, pyruvate oxidation, which is the conversion of pyruvate into acetyl-CoA, the Krebs cycle, which is the conversion of acetyl-CoA into carbon dioxide, NADH, FADH2, and ATP, and the electron transport chain and chemiosmosis, during which time the reducing power of the coenzymes is transferred to proteins that set up a proton gradient used to produce a whole lot more ATP. The amount of energy released by this process is equal to the amount of energy released by the combustion of glucose. So let's compute the total amount of energy produced by aerobic respiration. The theoretical yield of ATP during aerobic respiration is 38. The actual yield for eukaryotic cells is less than that due to many other biochemical reactions occurring simultaneously in the cell. A gain of 2 ATP occurs during glycolysis. 6 more ATP are generated from the 2 NADH produced during glycolysis. 6 more ATP are generated from the 2 NADH generated during the pyruvate oxidation. 2 ATP are generated during the Krebs cycle, while 18 are generated from the 6 NADH produced during the Krebs cycle. Another 4 ATP are generated from the 2 FADH2 that are produced during the Krebs cycle.
Anaerobic respiration is an ancient process that predates photosynthesis and the accumulation of oxygen gas on the surface of our planet. It consists of glycolysis and fermentation. anaerobic respiration only generates 2 ATPs per glucose. Aerobic respiration, on the other hand, consists of glycolysis, the oxidation of pyruvate, the Krebs cycle, the electron transport chain, and chemiosmosis. The final electron acceptor in the electron transport chain is oxygen gas. The proton gradient produced by the electron transport chain is used to produce a whole lot more ATP.